So host cells, what's mean host cells or host organism? Because we want to make sure that recombinant DNA molecule, to be able to be replicated. Sorry, to be replicated, because this recombinant DNA molecule are unable to replicate outside a cell. So in order for the replication to take place, the host cell must recognize the origins of replication. Okay, must recognize these origins of replication. So most of the times, okay, we can have many types of organism to help us, okay, to synthesize the protein, but usually E. coli we use. So why we choose E. coli in the genetic technology? Okay, so a few things that we can look at it here is, first, genetic simplicity, because they are prokaryotic cells, roughly with a very small genome size, roughly about 4,400 genes only. If you compare to the humans, about 30,000 genes. And the cells in the haploid state. Now, what's so important haploid state? Because we put in the gene of interest. If, let's say, it's a recessive one, then dominant allele will mask the effect of recessive. But bacteria, they only have one set, haploid. So therefore, they won't mask the effect of the gene expression. So their genetic is very simple. High growth rate. This is another one. Because we want more bacteria to produce. Right? We want more bacteria to produce the insulin for us. You cannot depend on one. You try to imagine the whole company only one worker. Definitely will be slow, the process. But if I have millions of workers, then I can have the more product form, right? Okay, so high growth rate. How high? If you look at this for E. coli, the replication rate after about 20 minutes to 40 minutes, one round of reproductions. Are you clear? So you can see that every 20 to 40 minutes, we double out the number of the bacteria. Okay, and bacteria, they grow faster when compared to the eukaryotic cells. Okay, roughly about one generation per 20 minutes to 40 minutes under typical growth conditions. So means that you will be able to see, you can get more bacteria that can produce insulin for us in 1 hour. Okay, let's say for us, our mitosis rate for humans under normal control is roughly about 24 hours, one round. If you look at the bacteria, every 20 minutes, one round. So definitely bacterial growth rate is very high, typically for the E. coli. Okay, about safety. E. coli is naturally found in our intestinal tract. So until so far, we know that most of the E. coli, they won't cause the disease to the host cells. Okay, and we are well studied about this E. coli because advanced knowledge of its protein expression mechanism makes it simple for us to use. We know how they produce protein. So simple, well studied, we can use it. And the most important thing is they can do foreign DNA hosting. So what's mean foreign DNA hosting? Basically means that they will be able to pick up. Okay, if you give them foreign DNA, they can express for us. Okay, and they need simple nutritional requirement. So it means that they are not fussy. Okay, you do not need to give them KFC, McDonald's, Pizza Hut, or whatever, because they just require the medium. Okay, the mediums, in this case, LB mediums is enough for them to grow already. They do not require a very, very sophisticated nutrients. If you look at eukaryotic cell that I deal with previously, okay, in my research, in my project, I use the human cells. So human cells, you need to give them growth hormone. You need to give them different kind of cytokine for them to grow because they require, and these growth hormones, they are not cheap. But E. coli, no problem, because they just require a very, very simple nutritional requirement. So because of these 6 factors here, why we choose E. coli as our host cells. Okay, so transformations, okay, what's mean transformation? Transformation means that it's a genetic alteration of the cell resulting from a direct uptake and incorporations of the exogenous genetic material from its surrounding. Now, what does this mean? Just now I draw this. If you have the plasmid with the gene of interest, so this one we call them as a recombinant plasmid. Okay, we call it as a recombinant plasmid. Now, if we have this E. coli, and this E. coli with the recombinant plasmid, they take up recombinant plasmid like this. So we know that these host cells, we say that the host cells of E. coli is transformed. Okay, like transformer here. Because why? Initially, the host cells cannot produce insulin. Now they can produce insulins already. So they are transformed. When host cell take in the recombinant plasmid, then we say that the host cell is transformed, because initially they can't produce. Initially, they can't produce the insulin, but now they can. But for these transformations to take place, the cell must be competent. So we're going to look at this, how to make the cell competent tomorrow. Okay, not all the bacterial cells can take. They must be in the competent stage first. Okay, so we are talking E. coli. It's so good to use E. coli because of the simple, nutritional, well studied, and blah blah blah. But there is, okay, or there are disadvantages of using prokaryotic host cells. Okay, so what is the problem if you use a prokaryotic host cells? So the lack of membrane-bound organelles such as RAF-ER and also Golgi apparatus found in the eukaryotic cells. So it means that they have what we call the very limited eukaryotic post-translational modification. So what is mean post-translational modification? We do know that, okay, I think I need this to show you guys in detail. Now, very simple, we do have a DNA, okay, our gene. Okay, we do learn this one in our protein synthesis. So we have the transcription process. Okay, after transcription process, okay, we form the mRNA. But this mRNA is an immature state. We call it a pre-mRNA. Okay, the process. This mRNA is a pre-mRNA, cannot be used yet. Okay. Then from the mRNA to become a matured mRNA that can be used, this one is called as the post-transcriptional modification. And this process only happened in eukaryotic cell. Prokaryotic cell, you will skip this. Prokaryotic cells will straight away. Okay, prokaryotic cell. I will tell you later what is the problem. Okay, after you get a matured mRNA, we're going to have a translation process. In the ribosome. So what we get here will be polypeptide. So this polypeptide have to go through post-translational modification. So, okay, also in this case require RAF-ER and also the Golgi apparatus. Both of them are not found. Both RAF-ER and Golgi apparatus are not found in the prokaryotic cell, only in eukaryotic cell. Before it can form the protein in precise three-dimensional shape. Okay, so what is this mean by post-translation modification? What are the process? First, folding. The polypeptide need to fold into the precise three-dimensional shape. Number two, if the polypeptides here or protein is a glycoprotein, we need to add, okay, short fragments of sugar. Okay, third, sometimes it's a quaternary protein. Means that consists of more than 2 polypeptide chains. How we join them together? So again, we do need the rough ER. So this thing, we need the rough ER to do it. Okay, we need the rough ER to do this. So without the RER, we won't be able, we won't be able to do the folding into the correct dimensional shapes, and also we won't be able to add short fragments of the sugar onto the polypeptides. We won't be able to form quaternary proteins. Okay, so now after we have proteins already, we need to do the secretion sometime. So in order to do the secretions, then we do need the Golgi apparatus, to transport them and carry out the exocytosis. Okay, so but the problem here is, if you look at the whole process, post-transcriptional modifications in eukaryotic cells and post-translation modification also in eukaryotic cells, these two processes are not or cannot be done by the prokaryotic cells. So main problem now come in. What's the main problem? If you take the insulin gene, this is from eukaryotes. So if from eukaryotic cell, you insert it into the plasmid and you put it into the E. coli. Too bad. Why? Because E. coli cannot carry out post-transcriptional. Modification. Oops, sorry. They cannot carry out post-translational modification. So I will start it with what's meant by post-transcriptional modification. Okay, this one actually in AS syllabus. Okay, most likely means you have gone through about this post-transcriptional modification. Now, what's with post-transcriptional modification? If you look at the gene in eukaryotic cell, gene in eukaryotic cell is a bit special. They may not be continuous one. If this is a gene, the gene consists of different segments. Okay, I label it segment 1, segment 2, segment 3, segment 4, segment 5. This, let's say, this insulin gene. So if the transcription process takes place, we get the mRNA. Okay, single-stranded mRNA. So this single-stranded mRNA also consists of 5 regions. Region 1, region 2, region 3, region 4, and region 5. Okay, so what will happen here is, in order to form the insulin, okay, let's say insulin polypeptide. In order to form insulin polypeptide, maybe region number 2 is not needed, region number 4 is not needed. Are you clear? Regions number 2 and region number 4 is not needed. So post-transcriptional process need to take place. Okay, before we can form this insulin polypeptide, we need to have the post-transcriptional modification. So what is the function of post-transcriptional modification? Basically, number 1, number 3, we need it. Number 5, we need it. So this 1, 3, and 5, we name it as, okay, coding region. Are you clear? We name it as the coding regions, or we call it exon. Okay, exon. Okay. Now, how about the blue color that we don't need? This one is called as a non-coding region. Means that we do not need to use it to code for our polypeptide. So non-coding region, or we name it as intron. Are you clear? Exons and introns. Okay. So what we're going to do here is post-transcription modification help us to remove intron and then join all coding regions together. So it forms the matched mRNA now. With region 1, region 3, region 5. And this process cannot be done. This process cannot be done by prokaryotic cells. Are you clear? Okay, so special about eukaryotic genes, as I say that, they contain what we call the coding regions and non-coding region. So what we're going to do here is, we have to remove the non-coding region first. If you give to the bacteria entire thing, bacteria are going to produce everything, and then it won't form insulin anymore. Because to form insulin, you do not need the region number 2, you do not need the regions number 4. We only need 1, 3, 5, for example. So we need to go through this post-transcriptional modification. So the process here, one of these, to remove the introns, means the non-coding regions, join all the coding regions together. So in this case, we will form what we call the matched mRNA. So this matched mRNA will be used. Okay, so no problem for eukaryotic cell because we used to do this. For us, we always do this. Day in, day out, every day, every moment, we do this. But prokaryotic cells, they cannot do this. So means that we need to think another way to do this already for bacterial. So means that for bacterial, we cannot straight away give the insulin gene. Cannot. If you give the insulin genes, they don't carry out the post-transcriptional modification, they won't code insulin for us because the insulin will have the 1, 2, 3, 4, 5. Cannot. Okay, so scientists think that another way to do this is we use matched mRNA. We use matched mRNA, we give matched mRNA. We start from matched mRNA. We don't want to start it with this DNA because DNA we cannot remove. DNA, we have no way to remove number 2 and number 4. We cannot cut it out. Number 2 and number 4, we want to cut it out, it's impossible, difficult, more tedious. So what we're going to do here is, because we know matched mRNA confirm only has coding region. So means that what we can do, we take matched mRNA, change it to DNA. And this DNA is known as cDNA, or we call it complementary DNA. So the process of taking DNA and make it into the mRNA, the process is known as transcription. Right. So the reverse now. We take the mRNA, make it into the cDNA. So this process is called reverse transcription. And the enzyme involved here is the reverse transcriptase. Okay, the enzyme involved here is reverse transcriptase. Okay, due to the limitations of E. coli. Are you clear? Okay, so let's look at this now. The notes. So we say that prokaryotic cells have limited eukaryotic post-translational modification. Protein expressed may not be able to correctly modify, misfold, or no glycosylations, and also difficult to facilitate the secretion of the expression of protein in high concentration because no Golgi apparatus. This one, no RAR. Okay, and also bacteria do not process. Mechanism for processing eukaryotic pre-mRNA molecules. So extra steps are needed too. We need to isolate the mRNA. This mRNA is the matched mRNA. Okay, from the eukaryotic cells and convert them into a double-stranded DNA. So we use the enzyme known as reverse transcriptase from the retrovirus, so that we synthesize the complementary DNA. So this cDNA now only have the coding fragments or exons. Sorry, just not spelled it wrongly. Okay, coding fragments or intron-free protein coding genetic information. So means that exon means coding region. If you forget in the exam, just name it as coding region. Intron means that non-coding. Are you clear? Exon, coding. Introns, non-coding. Okay, so it means that we need to remove introns. Only we can give it to the bacteria. If no, bacteria won't know. Bacteria will do everything. Are you clear? You don't ask them to interpret, oh, which one is coding, which one is non-coding. They don't know. Okay? So that's why we need to learn the last part here, the reverse transcriptase. Okay, reverse transcriptase. So a reverse transcriptase is an enzyme used to generate double-stranded complementary DNA. So the process is termed as reverse transcription. Just now I told you already. Okay, so what we're going to do here is, first you can see that this is mRNA. So using mRNA, we synthesize the first single-stranded DNA first. Can you see that? First single-stranded DNA. And then we use a DNA polymerase, use this newly synthesized one to make another strand. So at the end, we can form cDNA. Okay, so first the mixture of matured mRNA strand is extracted from the cells. We add the primer. So at this moment here, you may be confused. No worry, we're going to learn in PCR again, maybe on Wednesday. Okay, so a primer, don't worry about this one first. And then we use a deoxynucleotide triphosphates to make a complementary strand. From the red color mRNA, again, we get the blue color DNA. Then we're going to get these RNAs to denature the mRNA, and then we form a new one by using the DNA polymerase. We get a double-stranded cDNA. Okay, so this one for informations only. Okay, so we also can create a cDNA library from the mRNA because mRNA already spliced. Now, what does it mean splice? We join all the coding region together. Okay, we join all the coding region together, so cDNA produced lack of introns can be already expressed in the bacterial cells. So reverse transcriptase, we're going to use it in the insulin production because they no longer carry introns, so it won't translate. Okay? It won't be this intron will be translated using the bacterial ribosome. Okay, so with this, I have done for today.